So up next, we've got Dr. Roxana Myers. Roxana received her bachelor's degree in tropical horticulture from the University of Hawaii at Hilo and her PhD in plant pathology, specializing in nematology from the University of Hawaii at Manoa College of Tropical Agriculture and Human Resources. She's currently employed at the same location many of our presenters are today at the USDA Pacific Basin Ag Research Center, where her responsibilities focus. Get fired for that one. So welcome, Roxana.
Thank you.
Happy to have you.
Thank you so much, everyone, for being here this afternoon. I'd like to thank HFNA for this invitation. I'm always happy to share my research, and thank you for this beautiful lay. Today, I'm going to be talking about two different subjects. One is my normal topic, plant parasitic nematodes, and the second will be a new invasive insect pest that we have here in East Hawaii, the Queensland longhorn beetle. So this list of people here are, the team nematode, and they are the ones that are so crucial in helping me to complete all of this research. So I'd like to say thank you to them, first of all. So plant parasitic nematodes are microscopic, non-segmented roundworms, which cause billions of dollars of damage in agricultural crops worldwide. They have a stylet mouthpart, which differentiates them from other types of nematodes, and this is used to pierce the plant cells. So I had to throw in this photograph. These pictures were taken by Landon Wong on a scanning electron microscope. He went up to Virginia to work with a specialist to get these photos, and you can see the stylet in quite detail. And he was even able to extract the stylet and the esophageal pump to get this up close detail. So I thought those were great, and I just wanted to share that. So how do these tiny little organisms damage crops? They destroy and weaken the root system. They cause stunting of the plant. They interfere with the uptake of water and nutrients. And this all relates to making poor crop yields. So what can we do about this? Well, there's multiple things we can look at. One is prevention, cultural practices, host plant resistance, bionematicides, and chemical nematicides. So I always use this as my go-to slide, and this is the most important one. So I always keep it in here because if you don't have nematodes, you don't want them. But most people who don't have nematodes don't think about them. But it's really important to consider these if you're doing any type of farming. You need to propagate plant material by seeds or cuttings or tissue culture. Be sure to use sterile potting media, tools, and pots when you're working with nursery plants and grow them on clean benches. And have good sanitation practices. I've seen nurseries become contaminated when workers go from infested fields to clean greenhouses. So please be cautious of that. And also watch the water movement from infested beds into clean areas, especially downhill. Nematodes love to travel on water streams. So we have an integrated pest management program for burrowing nematode and anthurium. And of course, that starts with prevention. But in most cases, we have to look at other alternatives. One thing you can do is use plants that are resistant or tolerant to nematodes. And I thought John did an excellent job this morning talking about one of the long-term projects we have in developing this host plant resistance. Also, you can do eradication. You can use steam sterilization using a fabric hose covered by a rubber tarp to actually disinfest the growing beds. There's also hot water disinfestation. You can use, if you want to clean up your propagative material, it works very well with anthurium gobo. You can soak these gobos for 10 minutes in a 50C water bath. Be sure to put it into cold water immediately after to cool it off, because I have seen growers actually cook their material. So it needs to be cooled down immediately. That's very important. I saw a great technique where they laid the gobo horizontal and then all the new shoots came up, and it was just a beautiful way to start clean material for your farm. And there's also curative control. So you can use bionematicides or chemical nematicides. We've seen success in anthurium production using fluopyrium. We got 53% increase in flower production, as well as a 43% increase in leaf mass. So this chemical is very good at stimulating new root growth and new plant growth, which also helps the plant to overcome the nematode damage. But I know it is a little bit costly. So we're looking at other alternatives, and I'm very excited to talk to you about this new project that we just finished up. Is using ground papaya seed as a fumigant for plant parasitic nematodes. So this project was started by a University of Hawaii CTAHR student, Lauren Braley, who's working under Dr. Kunwi Wang in the department of the PEPS department. So her PhD has been focused on using this ground papaya seed as a biofumigant for foliar diseases of leafy vegetables. And she also looked at controlling root knot nematode. So I was so excited to hear some of her results. I told her, come out to Hilo, let's try this on burrowing nematode and Anthurium. So I got these slides from her because I wanted to share the results from this little trial. So thanks, Lauren, for sharing these slides with me so I can share them with industry. So as you know, papaya is a big industry here, especially in East Hawaii. It's a $6.7 million production every year with 740 acres in cultivation. But unfortunately, 30 to 50% of those papayas that are produced are discarded as culled papaya. And 30% of that culled papaya is seeds. So if we could find a use for the seed, it's a benefit both to our industry and also the papaya industry. So the interesting thing is the seeds contain a compound called benzoisothiocyanate. We just call it BITC, no H, okay. And what we do is if you grind this up, the chemical is released and we can use that as a biofumigant. So here's the process. We extract the seeds, then we clean them up, we dry them, we grind them in a mill, we use a couple different grinders, but it ends up being very fine with like a burr grinder at the end. So then we will take that ground papaya seed and we'll boil it in water with a little bit of tween as a surfactant. Then we'll cool it and then strain it. She started out using centrifugation, but in my lab, we just strained it with the cheesecloth. So it's a very easy process that anybody can do. And then we'd go out there and I just drench the plants. So we set this project up in the greenhouse at PBARC. And thank you very much to Greenpoint Nursery for providing us with some plant material to get this experiment started. We're very grateful for that. So we wanted to look at the ground papaya seed at 0.5% and 1%. And I just happen to have a neem product, Azagard, on hand, which is 3% azadirachtin. So I said, oh, let's just throw that in and we'll also take a look at that as well. And then we had a not amended with inoculated and then a non-amended uninoculated, just so we could compare the growth rates and the health of the plants. So we looked at root lesion, plant growth, and we extracted at the end of the trial, I think it was about 7 months, once a month drenching. And, forgot, at the very beginning, you have to put on the nematodes. So this is really interesting result. I was very excited about this because I tell you, in this many years that I have been working on nematicides, I found very, very few products that will actually be effective. Plant parasitic nematodes are really challenging to kill them. So I was over the top with these results. So let's look first at plant growth. We saw improved number of flowers with the 0.5% ground papaya seed. And we also saw an increase in plant height. So the number of leaves per pot looked good in both the 0.5 and 1%, but we were a little bit concerned about the other result and just wondering if maybe 1% might be a little bit too much, could be a little bit phytotoxic. So after looking at all the results, I'm thinking I'm going for 0.75 next time. So this is the root damage. And as you can see, both of the ground papaya seed treatments had a reduced root lesion rating. Okay. So this is the important part. How many nematodes do we get out of the anthuriums after this project was over. And you can see the number of nematodes was almost comparable to the uninoculated. We had a little splash. That's why there's that little bump on the end there. The results were excellent. And this was also backed up. We just finished an in vitro assay where we would take the nematodes and just put them directly into the solution of 0.5 and 1%. And so like the 1%, seeing that we got 100% mortality, and the 0.5 was around 30 to 40%. So we can see it both in the greenhouse and in the lab. So amazingly, this is like a fumigant. It's actually killing the nematode and not just increasing maybe the plant tolerance or stimulating the plant growth. So the neem was, it looks good. It stimulated some plant growth, but it wasn't that great in killing the nematodes. And in the in vitro assay, it also wasn't very good. But I just wanted to say, I want to bring up this other experiment that we did a while back, that not all neem products are created equally. So don't think I'm talking down on neem here. We did another study. We looked at another product called azasol, which has a 6% azadirachtin content compared to the 3%, and it's also formulated differently. So that's very important to understand how your neem product is made, what it's made from, and the process. So that makes a big difference. So you can see in this study was done on root knot nematode on tomato. The neem significantly decreased the number of nematodes in that project. So that's also worth a try. So if you tried it once and it didn't work, look at, analyzing another product. But overall, in the study that over the ground papaya seed, that was the winner for my experiment. So we're definitely going to move that into field trials next. So if anybody's interested in collaborating with that, oh, I see some hands. We'll talk later. Okay. So still talking about burrowing nematode, I just wanted to share this other project that I worked on. I was very fortunate to be asked to join this group to do a global comparative genomics project of burrowing nematode. And what they did was sequence and compared the genomes of 18 populations from 5 continents. So this was a lot of information and a lot of work. This manuscript has just been accepted, and we're talking like a 54-page manuscript. That's why there was, 54 authors on there, if you didn't notice. But it's a very important subject, and it took all of us to get this going, especially Catherine Ram, who headed up this project, and she works at the USDA ARS in Beltsville, Maryland. So burrowing nematode has been around for a while, but we really never did discover the origin of it. It was first reported in Fiji, but that doesn't necessarily mean that's where it comes from. But because it's grown, found on banana is propagated by suckers, and people love to share their cakey banana with everybody and take it to new lands as they go and travel. This is how it made its way all across the globe. Was on banana. So that was a big focus of the research, but they were also interested in what about these burrowing nematodes that are found on other crops besides banana? Are they related to this banana one? And that's where we came in. So we sent them some nematodes from an anthurium field that we had been raising in the laboratory. And then on our end, we did like a host range greenhouse assay, and we compared, we took our isolate of burrowing nematode, and we put it on anthurium, citrus, and banana. And then the colleague in Costa Rica, he did the same thing. And so the results were very interesting. Ours only went to anthurium. It didn't even go to banana or citrus, and his went to banana and had a very large reproduction, while ours, and he got a very, very small reproduction on anthurium, and a reproductive factor of below 1 means you get less nematodes off than you put in. So it behaved very, very poorly. So that raises a lot of questions. And there was also, there's I guess we'll call pathotype on citrus in Florida, which caused a very bad outbreak there. And this is the reason why we have this quarantine on us today, is because there's a big citrus industry in California, and they didn't want the burrowing nematode to get infected in their fields. but look, our nematode doesn't go to citrus. So it raises a lot of questions, and I think we can have a lot of conversations around this topic. Because when you look at when they did this relationship of the burrowing nematodes using DNA, they analyzed 116,000 what they call SNPs, which is just one nucleotide changes. And then they can come up with this phylogenetic tree, which shows the relationships of all these different isolates that they've collected, all over the world. So you can see the banana ones are all grouping together at the top, while ours, the anthurium and the citrus, are completely not related to anyone else. So we learned a lot with this study, and there's a lot more to be done. But I just wanted to share that because I thought it was super interesting. So let's switch gears and let's talk about Queensland longhorn beetle. So this is a large wood-boring cerambycid with a very wide host range. The larvae cause extensive structural damage, and it burrows inside the tree, and it completes its whole life cycle in the tree, just tunneling and feeding. So this is some symptoms that we will see on cacao. You see the sawdust-like frass, the sap, the lifting bark, the girdling of the trees, and then those round circles are the adult emergence holes. These are some other photos of citrus, kukui, and cacao. So you can see how destructive this pest really is. And for the ornamental industry, I'm sad to say, it's a severe pest on cycads. We've seen a lot of farmers with major infestations in East Hawaii, and it's a little difficult to see that bottom right photo, but that is a giant queen sago that has toppled over and is just mush on the ground. So we just reached our hand into the material, and then we grabbed that larvae and a bunch more. So they really like the cycad. It's very easy for them to chew on. It's also become a serious pest of quarantine importance now. These were cycads that were destined to go to the lanai. And let me tell you, this was a real hardship on the nursery because the inspectors at HDOA discovered that they were infested with the beetle. So of course, the shipment was off. So this is very devastating. We need to work on some kind of quarantine treatment now. And it's very sad to say that we have to deal with this. So I can't help right now with the quarantine part, but we are working with a beneficial nematode. Now, I know I'm always up here talking how bad nematodes are and I'm trying to kill them, but not this time. There's actually good nematodes that we can use to our benefit to try to help control these invasive insect pests. This nematode here was recovered from the island of Hawaii, Heteroditis indica, and we've actually found them throughout the state. It's very virulent. It's a broad host range. We've tested over 40 invasive insect pests. It usually works best with soil-borne pests. We've had the best result with sweet potato weevil. It's been very successful, but with this system, it's actually working quite well. And the reason it works so well is because this nematode is a hunter. It effectively navigates the galleries that caused by the larvae and will hunt them down, infect them, and then reproduce. And because these larvae are so large, we're getting like 200,000 to 400,000 new infective juveniles coming out of that one larvae. So once you make one application into the tree, once it finds one host, all of that nematodes children are going to come out and search for every living larvae left in that tree. And that's what makes this system work. So let me just show you real quick how to do the application. It's really important that you store your nematodes in a cool, dark location. They're very susceptible to UV light, but don't put them in the refrigerator because these are tropical nematodes and, they like it warm. We always apply the nematodes super early in the morning. And if it's pouring rain, that's even better because you want your nematodes to be fresh and you don't want anything, them to overheat, in any way during the process while you're in the field. Another very important thing is to agitate the nematode solution before you draw it up into the syringe because the nematodes tend to settle in the water very quickly. So if you want to make sure you have a uniform amount, every time we stir, then we draw it up in the syringe. And then get to work. So the first step before you even get your nematodes is to scout your fields very, very carefully. You need to look for all these damages that I mentioned, this oozing sap. And then what you want to do is you want to take, this is just Kathy, she's showing, okay, look, all the sap is coming out, and these are all the places where you're going to want to inject. So once we find this out in the field, we want to mark it with the flagging tape, each and every branch that you're going to treat. So that way, when you have the nematodes, you can just go out there and get to work, and you don't have to spend any time looking around, because it is very tedious to inspect every branch of every tree. But this is what we have to do now to be able to know, how badly this infestation is on our farms. So here's the injection process. Because in this situation, you're using hardwood trees. This could probably be different for cycads. I haven't done much work on infesting cycads yet. I'm really looking forward to working on that and seeing what we can do. But the important thing is with hardwoods, you have to find where the gallery is, or you're not going to be able to get that needle inside of the tree. So once you see all these soft spots, she's moving to all of the soft spots, injecting the needle, looking for those spongy places where it's eaten out underneath. And that's where, you're going to apply all the nematodes. And you have to really try to do it in every spot that you can, because you can only get so much solution into that gallery before it starts coming out, or it starts coming out the other side of the tree, the branch. So we really need to take the time. This is a very patient treatment technique. You have to have a little bit of patience. And I know it's tedious, but if you can do it successfully the first time, that season, you don't need to go back again. So it's not like you have to keep going back there all the time if you can catch it right away and do it correctly. So one of the field trials that we did was we did on avocado. So the first go round, we didn't have the great result, but second time around, we had 75% of the trees seemed to be cured. It's very hard to validate this type of experiment in the field without cutting down the tree completely. So what we do is we just look for any new frass, new shavings, new sap. And then if it kind of dries up and you can't find anything, oh, you got it. The nematodes did their job. And then later we'll go back and look for adult emergence holes. So at the end, after 3 months, one treatment a month, we got 85% cleared of the infestation. So with cacao, it was a different story with cacao. The field was heavily infested. So first time, 50% of the infestation was cleaned up. After that, we never could get a handle on it. It was so heavily infested. So it's really, really important to treat as early as possible, as soon as you see a sign. Especially right now is the season where the females have laid the eggs already. The larvae are still small under the bark. Now, this is a good time for the nematodes to catch up. The smaller, the closer they are, it's going to be easier. So try to treat as early as possible. So it's just super important to keep scouting your field and just being out there and familiar with all of your plants. So these nematodes are available to the community and to farmers at no charge. We have an agreement through BISC, the Big Island Invasive Species Council. So you can contact Jade Miyashiro or Darcy Yogi at BISC, and you can get on the list. And we're trying to get those out to you as soon as possible. We're doing some work here at PBARC where we're going to try to ramp up the rearing, that we can pass that on to BISC to make sure that, they can get those nematodes out right away when people need them, instead of having to wait. So that's what we're doing with the nematodes. And I'm very proud of these little guys, because they're out there, trying to fight this war with us. And we need all the help we can get. Okay, so I'd like to talk about one other trial. This is last topic, really fast. This trial is using the QLB females and trying to discourage the females from laying eggs under the bark. And I'm doing this in cooperation with Dr. Pascal Agbedian Atalar at the University of Hawaii, and he is a CTAHR expansion specialist in entomology. So this has been a really fun experiment if you like messing with giant beetles that scream at you when you you try to move them around. So we had a lot of fun. We got a lot of laughs and a lot of screams trying to do these trials, but we got through. So this hasn't been done in the field. Let me just tell you, this is the first time around for this experiment. All we did was use branches, kukui branches, just to simulate how this would work in the field. So we don't know how these products would go in the field, but I just wanted to share this because I think this information is very important for growers, because I hear a lot of people just trying stuff out there, and they have a lot of questions for me about it. So the things that we tested was latex paint, kaolin clay, hydrated lime with neem oil added, a 3-in-1 plant guard, and Daylin protective tree wrap. So I'm sure you probably haven't heard of the last two, so I just want to talk a little bit about that. The 3-in-1 plant guard is sold on the mainland. It's used as a protectant, a sunscreen, an insect repellent, rodent repellent. They had this little commercial where the rabbit's like, "Ooh, this tastes terrible," because it also has essential oils like castor oil and garlic oil. So I guess they find that repealing. So I got some samples and I figured we would add that to the trial. Then I also came across this Daylin protective tree wrap, which is a type of insect pest control. Insects can't get under it or lay eggs. It's a non-toxic, chemical-free alternative. But I have to say, everybody in my group laughed at me a lot about this, and I was just determined that I was going to throw it in as a treatment anyway, because I like the idea of it. They call it the pantyhose treatment. So, and well, I think it's more like an ace bandage, but here we are in the greenhouse. We're painting our logs, drying them, doing our wraps, getting it set up. So this is what one of the trials looked like. This was a choice trial. We would take every treatment and each log, there'd be all the choices in one box. And so we had this boxes and we had them in the screen container and then we had them in a screen insect building. So we're very careful not to let these females get anywhere. So we're doing like triple containment. Then we introduced 3 females to each box and see what happened. So in the top right, that was another trial we did, which was called a no choice trial. And we just put 1 treatment per box. And that's just to see, like, if they don't have any choice, what are they going to do? And then here you can see this is what it looked like. After a month or so, they're really chewing away. And some things they thought were quite delicious. As you can see, the little QLB female saying, "Man, this tastes yummy." And guess what treatment that was? The plant guard. So for some reason, those essential oils were great for a beetle. Maybe rodents or rabbits don't like it, but that sure did. So that was disappointing for me because I really liked the plant guard. It was very easy to apply with the paintbrush. And it smelled great. It smelled like peppermint and spearmint oil. So I was rooting for that one. Another one was the latex paint. A lot of growers I've heard have been using latex paint to try to protect the trunks of their trees. QLB love eating latex paint for some reason. It's very odd. So this is what the results look like. This is the no choice trial. We didn't see significant difference, but you can see, boy, they really love the plant guard. So this is the number of larvae and eggs that came out of each of these sticks. So what we do is we would split the logs open and we would count, hunt down all the eggs and count all the larvae. And so this is what this graph is showing right here. So for some reason, they even liked it better than no treatment. So that thing must be extra special. But we got some, much more clear results in the choice trial. This is trial 1. And you can see these ones with the A are the ones with the most larvae and eggs. So that's the control, latex paint, and plant guard. So the kaolin clay, lime and neem and wrap, those really looked promising. And same with trial 2, it looked about the same. The wrap, neem, and kaolin clay were the best. They did seem effective at discouraging the females to lay the eggs. And one thing, I should have said this at the beginning of the story, but the reason that this is so important about trying to deter them is because before the female lay the eggs, she will actually take a bite and she will see, is this a suitable host for me to lay my egg and for my baby to be in here, because that's where it's going to live, until it turns into an adult. So, taking that little taste, that's why I thought the wrap would be good, because it's just going to be like, what is this material? They're just going to move to the next tree, right? Or move to your neighbor's yard if you have all your stuff wrapped. So that's why I like the idea. But in conclusion, and this is, all preliminary, we haven't gone to the field, just so you know, the clay, the lime, and the tree wrap were the best. Latex paint and plant guard did not deter QLB at all. So I wouldn't recommend that. I'm still loving on the plant guard, so I'm thinking maybe I can use it as in the future and add neem to it. And I see Pascal's laughing because really the practical thing to do, which I should have put it on the slide, was to use the kaolin clay, because you can spray the kaolin clay with a pump sprayer, and it worked just as good as all this stuff, and it's probably less expensive. An important comment, I just want to make sure you do not mix the neem oil with the paint, right? You can't mix water and oil, but also it will cause a reaction, which will be dangerous for your tree. It could actually harm your tree. So please don't do that. But finally, I wanted to show off my beautiful wrapping job that I did on this poor avocado tree. Because my group was like, really, you think people are going to do this? And they made me go out there and do it by myself while they watched and laughed. So it took me 2 rolls of wrap and about $10 worth. And I only could do like the main trunks, right? So you can't do like the top branches, but at least you're protecting the trunk, because once the larvae get in the trunk, that's where you can actually get death of the tree, because it's going to like girdle that trunk. So I figured, you can watch for infestations on the top and either treat them with the nematodes or cut them off. So at least you're protecting the important points. But of course, if they get in the top and you don't pay attention, they can still go down to the trunk. Okay, so I did leave that on a couple months because I have to wrap it really tight, and I was a little bit worried about the breathability and, is that going to cause any problems on the tree? But when we removed it, it looks great. So that's still an option for anybody who's interested, but probably if you want to start this spring before the females come out, and you want to, try something. I'm not sure how long the kaolin clay will hold up in Hilo rain, but if you're determined to try something, that would probably be my recommendation, and we'll be doing some field trials next spring. So thank you to everyone who helps with these projects, and doing research is definitely a team effort. I do tons of work with the UH-CTAHR, which is my alma mater too. So it's great working with my colleagues and classmates, and I'm really happy that they'll join me in doing this research. So thank you so much to everyone, and appreciate it.
So unfortunately, as moderator, I have to be the cruel timekeeper. And Eric and I shook hands for too long, so we don't have too much time for questions. Other than I'll give Tessie one question that she had online, and then the rest of you in person can ask as we go along. So I'll allow for one question. And Tessie was asking, what happens after the predatory nematodes, the parasitic nematodes, have eaten all the larvae in the trees?
Okay.
Yeah.
So the nematodes can survive for a few months without a host. So that's good. So they can just wait it out. They will just go kind of into like a static state, and then they will just wait. And as soon as they get, like they sense a new host, then they'll wake back up and go towards it. But there's also a lot of different insects inside the tree. So especially when you have Queensland longhorn beetle, what we're seeing is a lot of ambrosia beetles will come in. So once the tree is wounded, it sends off these signals. You get other type of secondary insects that come in. So in the meantime, hopefully the nematodes can feed off of these smaller beetles until another egg is laid and the process starts again. But usually I would think every season you would probably want to start a fresh treatment again, just to be safe.
Great. Thank you. Okay. Next we've got my good friend Hannah Luchin, who's also my counterpart on Maui County. Hannah is, as I just said, the Maui County Floriculture and Landscape Extension faculty